Hi, I decided to record a short lecture about how bioinformatics pipelines work, and as an example, I will talk about uh, evaluation of NGS targeted pipelines used in oncology, with example of lymphoma for MRD testing in and how to orient it in cell-free DNA. So this will be aimed to work with minimal amounts of DNA molecules in blood. So what do we need to have to run such a pipeline? We will have to use a lot of computational resources. It will require high performance CPU nodes or cloud instances to process all this data gathered from sequencing. It will require more than 32 gigabytes of memory to process these large genomes and large files which we have as an input. And of course we will need to store this data somewhere, and it's better to store on fast working hard drives. As a data input, we will work with raw sequence files, FASTQ files, and we will have to use reference genome for alignment and how our reads correspond with this genome will be necessary for us for this process. Also, as we are using target panel, we will need to have this panel definitions. They should be structured in a bed file. I will talk about general tools which are publicly available, how they could be applied in this pipeline. So I will name all of them lately. We'll need to have some stuff for preprocessing in trimming and UMI handling. Also, we need tools to align these reads with our reference genome. In the next stages, we'll need variant calling packages and packages for metrics and quality control. And of course, we need to organize this process in a step-by-step pipeline, and in most of the cases it's being done with tools like Nextflow. Also, we can use containerization like Docker to use packages which have all the dependencies in controlled and shareable package. It helps with version control and distribution of these packages in our environment. For quality control, we'll use data provided in our run as a metadata. Also, we can use a reference standard, which could be added to our samples, so we can make this validation. And of course, we'll need this process to be tailored, documented, and we need our samples to be easily identified in future to process in this process. I will not focus on overall workflow, but I will just highlight where is the place of bioinformatic tools in this process. So we have stages for extraction and preparation of this DNA. There are additional steps to extract cell-free DNA if we need to work with these sequences, and after that we'll need to prepare our library by adding unique identifiers for molecules, barcodes, and after that, during sequencing, we will be able to have our reads in a certain length and certain depth. I will cover a bit later what stand for. And after that, when we have our reads, we can process them with our bioinformatics pipeline. So in this process, we will need to trim these adapters containing barcodes, identifiers, and so on. We will need to use the duplication tools to select best reads, which will tell us what's the real sequence of nucleotides in this DNA. After that, we'll need alignment tools like BWA and error suppression tools like UMI consequence tools. This will result in a sequence of nucleotides where we're sure which one stand after another, and we can use this data for variant calling. There are different tools for this stage, and after we have our VCF files, we can use annotation and filtering to focus on most interesting areas of genome, which one we are going to work with. And after that, we're able to process this data to produce our outputting results with for each sample, providing annotated data about these reads. What interesting for bioinformaticians steps are in library preparation. It will be addition of unique molecular identifiers, which are random sequences, which they are added before amplification of our DNA sample, and they will allow us to connect different reads which are related to a certain initial molecule, initial part of DNA. And it could be used later on error correction stage, where we will be able to select best reads from the whole amount of multiple reads that we have. And these libraries of UMI contain millions of unique DNA fragments, and they can efficiently be used to identify each molecule in our sample set. After that, we are using sample barcodes to identify certain samples through the whole reads, so we can combine multiple samples in one library and later understand which reads are corresponding to which sample in this library. So combining together these molecules resulting in so-called adapters. They're used for binding to flow cells, so this process for sequencing will be possible. There will be sequencing primer sites. They contain known sequences of the primers that are used for sequencing. There are indexing barcodes to identify specific samples in a library, and UMI embeddings that are used for identification of unique molecules in our samples for error correction and demultiplication of the data. Sequencing depth or coverage correspond to number of times a nucleotide base is read during sequencing. So if we are working with low allele frequency, like in cell-free DNA or MRD analysis, we will need to have as more depth as possible. So we'll be able to filter out our regions of interest from our resulting reads. Biological samples often yield tiny amounts of DNA, so for most of the sequencers will need to provide amplification of this data, and this will allow us to produce strong enough signal for accurate base calling. And also these reads are being provided by bridges forming clusters in these flow cells, which allow sequencers like Illumina to make these reads. And when we're working with different types of samples, we will need to think of different required depth of these reads, because if we have ultra low amount of initial DNA molecules which we need to focus on, like in cell-free DNA, we will need to cover them with much more depth. So getting back to our pipeline for bioinformatics data, we'll start with adapter trimming. There are artificial additions to our biological samples, so we don't need them for alignment, and if we will keep them, they can result in incorrectly mapped reads, and also it will result in bigger files, so we are able to delete them, trim them, and discard. For example, it could be done with a tool called Cut Adapt, where we're just placing our sequences and our FASTQ file to be to apply this trimming. But before that, we'll need to store these UMI identifiers somehow, and we can use tools like UMI tools package, which allow to extract this UMI sequence and add it as a tag in a resulting file for each read. As a next step, we can use the duplication. It will allow us to group reads by mapping certain UMI tags together, and these groups share same chromosomes, same sequence in initial nucleotides, and they could be used for consequence analysis, and the resulting file will represent highest quality reads from the same molecule from all reads in our library. We have different tools to make it possible, like FGBio or UMI tools, and they will align all reads in the UMI group to each other, so they are connected by the UMI adapters, and at each reference position it will count bases across all reads, weighting by quality scores, so we're able to understand which reads are more precise, and applying different thresholds we will be able to select a minimal level of certainty that these reads were made correctly, and to result in consequence file with duplicated information from initial reads. We are always working with different options and configurations in our tools, which could be adjusted to a certain sample type or initial data quality or specific library preparation kits and so on. So for example, we're able to correct how accurate UMI reads are, and for example, if there is a one error in UMI region, they are still able to be connected together and used in this duplication stage. Also, we can state, for example, minimum amount of reads which should be present for this consequence to be calculated, and what's estimated error rate which we're accepting in this process. Another improvement which could be applied here is base quality score recalibration. It could be used to adjust per base quality scores to correct known systematic errors like machine-generated cycles, sequencing context, and they're being used to improve accuracy of variant calls in the end. So we have, for example, these capabilities in JTK toolkit like base recalibrator, and they could be applied here. Now we're able to proceed with alignment, and on this stage we're aligning extracted reads to the reference genome. So we need to have our reference genome already ready to use these tools. For example, we can use HD 37 or 38, and we also have different settings for these tools. For example, in PWA-MEM tool, we can set up minimal size of these fragments of DNA, and for example, we can correct it for shorter reads. For example, if our samples are damaged or we need to align them with a certain purpose. Also, we can increase scores which could be used as a result in this alignment, and this also could calibrate our process a bit in one way or another. So as a result, we will convert our FASTQ data, which is exactly a certain read with a sequence of nucleotides, to SAM file, which is a human readable BAM file, and in here we'll have connection between our read and genome position where this read aligns to contain chromosome, its position, level of confidence, and how many base pairs are matching to this fragment. Next stage is variant calling. We're using software that examines mapped sequence reads against a reference genome to identify differences, single nucleotide variants, insertions, deletions, and structural variants. These variant callers are using statistical models and filters to distinguish true variants from sequencing or alignment errors. We have multiple tools here, and some of them could be used to compare germline and tumor samples. There are haplotype caller, prebias, MyTech, Lowfrag, Vardict, SIN-Vict, and so on. Different variant callers have their own strengths and limitations, and for our purpose most interesting will be verdict, as its focus on low variant calls, and another one, SINVICT. It's also quite preferable for low and ultra low VAF in cell-free DNA, and it will require some tweaks to make possible to use it. And at the same time, it's totally possible to use multiple different variant colors and compare the results together to process different approaches. For example, you can use MUTEC2 in tumor-only mode to call somatic variants in tumor DNA when no much normal DNA is available. Also, we can configure verdict to catch ultra-low frequency variants also. It will require us to provide reference genome, FASTA file, and verdict will use this to align interpret read mappings. We can set allele frequency threshold to 0.1%. Also, we'll need to name our samples, provide BAM files with deduplicated alignment results, and we can combine this tool with filtering stages, for example, to use removing tool which will reduce reads variants which are on only forward or reverse strands, and they are likely to be artifacts. Another interesting option will be to use sensitive nucleotide variant calling, and it's tailored for ultra-sensitive detection of single nucleotide variants and small indels in circulating tumor DNA. It uses a probabilistic model that accounts for sequencing error rates and local sequence context to distinguish true variants from noise. It applies trend and position-based filters internally to reduce false positive without an external tooling to be added in this tool. So it's also a good idea to use this. Once variants are called, the next step is to interpret their potential impact and remove likely artificial ones. For example, we can use a tool from JTK library which will remove variants which were called as artifacts or low confidence variants or germline variants. We can go deeper and use functional annotation with WEP or SNP-F, and these tools will add gene symbols and include information how this nucleotide will result in protein changes. Different types of samples have different artifacts, and there are tools available which are able to use blacklists of known artifact size or context-based filters which could be applied for our on our VCF file. For example, VCF tool could use curated blacklist filtering for this task. There are additional tools which could be used for contextual filtering, and as a result we should have annotated VCF file covering k-info and format data related to our sample and for each variant allele frequency we need to have proportion of reads supporting the alternate allele and total number of reads covering the variant size and number of unique UMI which are related to these alternate alleles. For example, in our resulting VCF files, it could look like this, and we have a number of reads supporting this variant and total depth in this position and number of unique UMIs observed with the alternated allele supporting that there are, for example, 20 reads derived from 18 original molecules here. In residual disease monitoring, we will need to associate these detected variants with certain time points, and this will be stated in our report how these variants are connected with the certain stages of the monitoring process. For example, such a report could look like this. So we have different genes associated with certain disease and mutations, and we state we are tracking number of molecules with a certain mutation which we were able to detect with a certain level of confidence in these samples. So talking about how well our pipeline is working, we need to analyze its coverage. It will be the fraction of targeted regions covered at above than 20% of the mean sequencing depth. So it will ensure that every hotspot or exon in our panel is sequenced deeply enough for reliable variant detection, avoiding blind spots. Quite close to that is on-target rate, which will constitute the proportion of total sequenced reads that align within your defined panel regions. So higher on-target rate is, the higher efficiency of capture for our regions of interest are in our results. Duplication rate will correspond to the percentage of UMI groups flagged as duplicates, and if we have low duplication, this will mean that we're sequencing unique molecules rather than amplified fragments of one molecule which was in initial sample. Also, we can monitor specific artifacts which correspond to our sample if we have a list of known invariant sites for this sample. We can use it to monitor error rate in reading this variant. Variant allele frequency will be related to coefficient of variations of measurements across technical replicates of the same sample. Consistent variant allele frequency is quite necessary for minimal residual disease monitoring, as high variability here will reduce our confidence in small changes which are happening over time. And the last one to name will be limit of detection, which will correspond to the lowest allele frequency at which the assay detects a variant with 95% sensitivity. And it will define our pipeline capability to call rare variants, which are quite an interest for us in such a project like cell-free DNA detection.